Hello students. Students, today we will start a very new topic, that is transcription. Let's see what is transcription. Transcription is the process of copying genetic information from one strand of DNA into messenger RNA. So the process of copying genetic information from one strand of DNA into messenger RNA is known as transcription. And with the help of information present in messenger RNA, polypeptide is synthesized, which is known as translation. In today's topic, we will discuss only transcription. So, in a dsDNA, that is double-stranded DNA, there are two strands. Suppose this is your strand which has polarity 3' to 5'. The strand having polarity 3' to 5' will be called as template strand. It is also called as antisense strand. The other strand which have polarity 5' to 3', it is called as the coding strand or sense strand. So here students, you can see that in dsDNA there are two strands, template strand and coding strand. As I told you earlier, the genetic information will flow or the genetic information will be copied from one of the strand, and that strand from where the genetic information is copied, that is the template strand. Now here the enzyme which is required for this process, or we can say the main enzyme which is required for the process of transcription, it is RNA polymerase. This RNA polymerase is the main enzyme for the process of transcription, and this process of transcription, it is taking place in the nucleus of eukaryotic cell. So, what we have learned? We have learned that the transcription is the process of copying genetic information from one of the strand into messenger RNA. Only one strand will participate in this process, and that strand will be the template strand having the polarity 3' to 5'. The main enzyme for the process of transcription is RNA polymerase, and in eukaryotes it takes place in the nucleus. Now we will discuss about the RNA polymerase. RNA polymerase is a complex enzyme. And it have three important parts. Let's see what are the three important parts. The first one is the core enzyme. This core enzyme contains 4 subunits, 2 alpha subunit, 1 beta subunit, 1 beta dash subunit. All these subunits are always united together and they constitute the core enzyme. Now let's see what is the function of these subunits. Alpha subunit. This alpha subunit is required to bind regulatory protein. the beta subunit, it is required to bind to DNA template. And the beta dash subunit, it is required to bind RNA nucleotide. So here we can see the first part of RNA polymerase is core enzyme, which is made up of 4 subunits, 2 alpha subunit, 1 beta subunit and 1 beta dash subunits. These subunits are always present together. The second part is sigma factor. Sigma factor is required for the recognition of promoter site, promoter is that region from where the process of transcription will start. The RNA polymerase will bind to the promoter site. And the second thing is, it is required for initiation of transcription. Initiation of transcription means to start the process of transcription. It is not binded to RNA polymerase every time. It will bind only and only when the process of transcription is to be started or for the initiation of transcription. And the third part which is required is the rho factor. This rho factor, it is required for the termination of transcription. When the process is to be stopped, it is to be terminate, what happens? This rho factor will bind to the RNA polymerase. So we can see that the RNA polymerase, it is a complex enzyme having 3 important parts: core enzyme, sigma factor and rho factor. My dear students, you should always remember that in the case of prokaryotes, in prokaryotic cells or in prokaryotes, there is only one type of RNA polymerase. And this RNA polymerase will catalyze, or we can say will transcribe, all the 3 types of RNA, that is rRNA, ribosomal RNA, tRNA, that is transfer RNA, and mRNA, that is messenger RNA. So in prokaryotes, there is only one type of RNA polymerase, but in eukaryotes, there are 3 types of RNA polymerase. RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. This RNA polymerase 1 will transcribe rRNA, that is ribosomal RNA. RNA polymerase 2 will transcribe your hnRNA, that is heterogeneous nuclear RNA, which will later lead to the formation of mRNA. And RNA polymerase 3 will transcribe tRNA, that is transfer RNA. So here we can see that in prokaryotes there is only one type of RNA polymerase which will transcribe all the type of your RNA. But in case of eukaryotes, there are 3 type of RNA polymerase: RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. So in this lecture we have studied about RNA polymerase and a little information on the process of transcription. In my next video we will discuss about the transcription unit. Thank you. Hello students. Students, in my last video we discussed about RNA polymerase, and today we will study about transcription unit. This topic is also very important from the exam point of view. So let's see what is a transcription unit is. transcription unit is a sequence of nucleotides in DNA that codes for single RNA molecule. This transcription unit contains three important parts. So let's see what are the three important sites or three important regions. The first one is a promoter region or promoter site. Second is the structural gene. And the third one is a terminator. So here we can see a transcription unit. It contains a promoter region, a structural gene and a terminator region. First of all, we will draw the structure of a transcription unit. As I told you earlier that in a dsDNA, that is double-stranded DNA, there are two strands. The strand which having polarity 3' to 5', it is known as template strand. And the other strand which has polarity 5' to 3', it is known as coding strand. So in a double-stranded DNA there are two strands, template strand and the coding strand. Next important thing is promoter. It has a promoter. This is the promoter. And here is present the terminator. In between the promoter and terminator is present the structural gene. As we discussed, a transcription unit, it contains three parts or three regions: promoter, structural gene and the terminator. Now we will discuss all these three sites in detail. First of all, a promoter. You should know the location of the promoter site. Promoter is present at the 5' end of the coding strand. Whenever you are talking about the location, you have to take the reference of the coding strand and not the template strand. So here we can see that the promoter is the region which is present at the 5' end of the coding strand. So where it is present? It is present at the 5' end of coding strand, or we can also call it as upstream of structural gene. Now second important thing, what happens to the promoter? The RNA polymerase will bind to the promoter. RNA polymerase is the main enzyme, it is the main hero which is required for the process of transcription. So this RNA polymerase will bind to the promoter and it will start the process of transcription. So what happens here? RNA polymerase binds to the promoter site. But to recognize the promoter, it needs the sigma factor, which is not always present with the RNA polymerase. So the sigma factor will come, it will bind to the RNA polymerase, and with the help of sigma factor, what will happen? It will recognize the promoter region. And sigma factor will also help in the initiation of the transcription. So the important thing is that RNA polymerase will bind to the promoter, first thing. Second, it requires the sigma factor for the recognition of the promoter site and to start the process of transcription. In prokaryotes, we see that this promoter has specific AT-rich region, and this AT-rich region in the prokaryotes is known as your Pribno box. It means that in the promoter there is present a sequence of 6 base pairs, that is TA, TA, AT, which is known as Pribno box. But if we see in the case of eukaryotes, there is also present AT rich region in the promoter, which is known as Tata box. It is also called as Hogness box. And it has sequence of 7 base pair, that is TA, 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 T. Here you can see 1, 2, 3, 4, 5, 6, 7, or TA, TA, A. Clear? So here we can see that in prokaryotes there is some specific AT rich region present in the promoter, which is known as Pribno box, having 6 base pairs, and in eukaryotes it is known as Tata box or Hogness box. Next is terminator. Terminator is that site or that region which will stop the process of transcription. When the RNA polymerase reaches the terminator, what happens? The Rho factor will come and it will bind to the RNA polymerase and it will stop the process of transcription, as I told you earlier in my previous video. So terminator is that site where the RNA polymerase will reach and the Rho factor will bind to it and it will stop the process of transcription. This Rho factor is actually a type of protein which is required for the termination of transcription. Next is the area between the promoter and the terminator. Clear? That is known as the structural gene. So now we will discuss about the structural gene. Structural gene it is the segment of DNA, or we can say the area of DNA that is required for the transcription and which will help in the process of formation of messenger RNA. This structural gene is of 2 types, monocistronic and polycistronic. Before going to monocistronic and polycistronic, you should know the meaning of cistron. Cistron, it is the segment of DNA coding for polypeptide. Very, very important. What is cistron? It is the segment of DNA which is coding for the polypeptide. Monocistronic structural gene means it contains only 1 cistron. Polycistronic means the structural gene contains many cistron. Again, the structural gene is of 2 types, monocistronic and polycistronic. Monocistronic means it contains only 1 cistron. Polycistronic means it contains many cistron. So here what will happen? If there is only 1 cistron, so it will carry information for one polypeptide. Here many cistron, so here they will carry information for many polypeptides. This monocistronic structural gene, it is present in eukaryotes. And polycistronic structural gene, it is present in prokaryotes. So here we can see the structural gene. It is the segment of, or we can say the area of DNA which is required for the process of transcription, and this structural gene can be monocistronic or it can be polycistronic. Monocistronic contains one cistron, polycistronic contains many cistron, and this monocistronic is present in eukaryotes while the polycistronic is present in prokaryotes. One more important thing is that in eukaryotes, the genes are split genes. Now what are these split genes? Split genes are those genes which contains the coding as well as non-coding parts. Coding parts are known as exons and non-coding parts are known as introns. So here in eukaryotes we can see that both the coding and non-coding parts are present, and that's why that type of gene is known as split genes. When the messenger RNA is synthesized or formed, what happens? It contains only the coding parts while the non-coding parts are not present. So this was all about in today's video. In next video we will study about how the process of transcription takes place. Thank you. Hello students. Students, in my previous video we discussed about the transcription unit, and now today we will discuss about the whole process of transcription. As I told you earlier that the transcription is the process of copying the genetic information from one strand of DNA into messenger RNA. This transcription actually consists of three important steps, and we will see what are those three important steps. So the three important steps are, first is initiation, second is elongation, and the third one is termination. So the three important steps of transcription are initiation, elongation and termination. Here we can see a DNA helix. This DNA helix has two strands, a template strand having the polarity 3' to 5' and a coding strand having polarity 5' to 3'. In between the two strands there is the hydrogen bonds present. So what happens when the process is to be initiated or when the process of transcription is to be started? The RNA polymerase, it will bind with the sigma factor. The sigma factor will come and it will bind to the RNA polymerase. This sigma factor, as I told you earlier, it helps us to recognize the promoter site. Second important thing, the process of transcription will be initiated only and only when the sigma factor will bind to the RNA polymerase. So, in this promoter site, what will happen? The RNA polymerase will bind and now it will break the hydrogen bond. The second step is elongation. During this process, what happens? When this RNA polymerase moves, it will break the hydrogen bonds and the ribonucleotides. Remember, ribonucleotides, because we have to synthesize RNA. So ribonucleotides, which are present in the form of triphosphate, they will come and they will lie opposite to the template strand. This is our template strand which has been shown with the red color. So what will happen? The ribonucleotides, they will come and they will lie opposite to the template strand or antisense strand. I told you it is present in the form of triphosphate, that is ATP, GTP, CTP and UTP. Clear? Now this RNA polymerase, what it will do? It will form the phosphodiester bond between them. So during elongation, RNA polymerase, it move along the template. Second thing, the ribonucleotides, they will come and they will lie opposite to the template strand, and a phosphodiester bond will be formed between them. My dear student, one thing you have to remember, this process of transcription requires energy, and how does this energy will be met during this process? For that, an enzyme, remember, pyrophosphatase. This enzyme hydrolyzes, it will hydrolyze 2-phosphate from triphosphate. And when it will hydrolyze 2-phosphate, what will happen? Energy will be released, and that energy will be used for the process of transcription. The third step is termination. RNA polymerase will move and it will reach to the terminator site. Terminator site is that site where the process of transcription will stop. And to stop it, it requires a factor, and that is our Rho factor. In case of prokaryotes, Rho factor is required for the process of termination, and this Rho factor is actually a type of specific protein. Now, what will happen? At the terminator site, the RNA polymerase will be removed, the Rho factor will also detach, and we will get the RNA. One thing you have to remember, the whole process of transcription in the case of prokaryotes, it is taking place in the cytosol. But in eukaryotes, it takes place inside the nucleus. One more important thing, the RNA which is being formed, or the transcription process is taking place in the cytosol, and in the same time the process of translation also starts in the case of prokaryotes. Clear? So this was all about transcription in prokaryotes, and now we will discuss the process of transcription in eukaryotes. Students, now we will discuss transcription in eukaryotes. In eukaryotes, the process of transcription takes place in the nucleus, while in prokaryotes, it takes place in the cytosol. Now, after the process of termination in eukaryotes, we got the hnRNA. hnRNA is heterogeneous nuclear RNA. This hnRNA has certain coding and non-coding sequences. The coding sequences are known as exons, and the non-coding sequences are known as introns. So to get the messenger RNA, there is processing of hnRNA. Now when we do the processing of hnRNA, it includes various steps, that is capping, tailing, splicing, and ligation. So let's see what are these capping, tailing, splicing, and ligation is. So as I told you that we got the hnRNA. This hnRNA is very unstable. So what we do is, at the 5' end of this hnRNA, we add a cap-like structure which is made up of methyl guanosine triphosphate. Do remember, at the 5' end, we add methyl guanosine triphosphate and it forms a cap-like structure. Similarly, at the 3' end, we add 200 to 300 nucleotides of adenylate acid. And it is known as poly-A tail. So what we have done? We have done capping and tailing. Capping is done at the 5' end, where methyl guanosine triphosphate is added, and at the 3' end we add 200 to 300 nucleotides of adenylate acid, which is known as poly-A-tail. After that, what we do, that the introns is removed with the help of an enzyme called spliceosome. Now, what is this spliceosome? Just see here, there are certain proteins required for this, and these certain proteins are called as SNRPs. SNRP stands for small nuclear ribonucleoprotein. These protein along with some other protein forms a complex, and that complex is known as spliceosome. So what does spliceosome do? It do the splicing. It cut and it removes the non-coding sequence, that is introns. And after that, what happens? We have only the coding sequences, and these are attached with the help of the enzyme ligase. So that is ligation. After that, we will get our messenger RNA. So the process of transcription is over here. The messenger RNA is synthesized, and this messenger RNA will come out of the nucleus and it will go to the cytoplasm. So this was all about the process of transcription.